I think the screen is shared. Can you see the screen? Yeah, it's shared. Hold on, guys. So going back to the replication, so we already know that what are the important proteins and enzymes that are, you know, that participate in the process of replication. Correct, right? So if you have any technical issues or any specific issues, please text so that I need not ask you again and again, and we can continue with the class, right? Which is quite important right now. So what happened in replication? So last class, the reason why we could not get the proper grip on the topic is because initially I was trying to explain you guys what are the important factors that are involved in the replication and what is their role. And after understanding the role, the mechanism becomes quite simple, correct, right? So that's what my thought is. But unfortunately, hold on, I will just try to insert one new slide. So hold on one second, guys. Uh, let me take this slide and yeah, I hope you can see me. So what we're discussing is the components involved in the replication process, right? So what happened? There are two strands which we are very much aware, and these are called as parent strands, correct? So if you see the mechanism of replication, which is a quick, I will do a quick revision, guys, so you will get all the points that we want to understand. Got it? So we know that the parent strands have two strands, and one strand goes from 3 prime to 5 prime direction, and other strand goes from 5 prime to 3 prime direction. So we are very much aware of the polarity of our strand, correct, right? So we know what is polarity and why the strands are antipolar and everything, correct? So this we don't have doubt. Now what happens when the replication starts? The replications happen on both the strands, guys. So we know that we have two strands, sorry, we have two strands, correct? Two parent strands. Both the parent strands are involved in replication. That means this is a parent strand, this is a parent strand, and this strand helps in synthesis of one daughter strand, and this strand helps in synthesis of another daughter strand. So each parent strand will divide or separate to become new DNA, right? That is what we learned in semi-conservative mode of replication. That means for every new DNA that we're going to produce, we have one parent strand and one daughter strand, correct, right? So we will have one parent and one daughter strand. So this is how the replication happens. But if you look at the total mechanics, like how the entire process of replication happens, we know that there is a difference in each strand on how they replicate. See, if the daughter strand has to be synthesized, let's assume this is our daughter strand, and every daughter strand is synthesized in the same direction. Which direction? 5 prime to 3 prime direction. Why always replication happens from 5 prime to 3 prime direction? It's very, very simple. So that goes with a very important polymerase that we see, that is called as DNA polymerase, correct? No? Our DNA polymerase. So our DNA polymerase enzyme is a DNA dependent DNA polymerase, we call it as. So this particular enzyme requires, okay, a compulsory 3 prime OH. I told you what is a compulsory 3 prime OH. Means, for example, our DNA is made up of nucleotides, and the nucleotides have what? A pentose sugar. In pentose sugar, we know that there are 5 carbons, like 1, 2, 3, 4, and one more is here. Five carbon atoms are there, correct? In that, at the third position of this carbon atom, what is present? OH group is present. And we know that in the fifth carbon atom, what is present? A phosphate group is present. Got it? No. So now if we take two nucleotides, okay, which we have to stitch together to form a DNA chain, in the third place, what do we have? We have a OH. In the fifth place, what do we have? We have a phosphate group. Usually, not usually, all the time, from one nucleotide, let's say that this is one nucleotide, this is another nucleotide. Okay. So if we have to stitch between them, or if the DNA polymerase have to make the chain, what do? They take the phosphate, they take the OH, phosphate of one nucleotide, OH of another nucleotide, and they create what? They create a phosphodiester linkage so that a chain of DNA is established. or you can also call it as a polynucleotide chain is established. So for that process, what do they require? They require a free 3' OH. You see how the OH is used. So our DNA polymerase do not have the capacity to synthesize the DNA from the first base pair. Okay, from base pair 1, they cannot start the DNA synthesis. So what do they take the help of? They take the help of primers. Correct? No, they take the help of primer. So a primer is what? A primer is something that can start the DNA replication from the first base pair. Okay, it has the capacity to start. It does not require a free 3' OH. It can lay the first nucleotide on its own. So what is our primer? Our primer is DNA dependent RNA polymerase. So you can understand what an RNA polymerase can do. RNA polymerase synthesizes what, guys? It synthesizes a RNA. So, sir, why are we synthesizing RNA in DNA synthesis? Simple. This one will not synthesize a very long RNA, but a very short RNA, which is around 10 base pairs. Why is the reason? Imagine an RNA sequence of 10 base pairs here, guys. Okay, what is left here? A 3' end of the RNA. So what do we get at the 3' end? A OH group is present. Correct, no? A OH group is present. So now what did we understand? The DNA synthesis happened on the both the strands. For the DNA synthesis, always the direction is from 5' to 3' direction. Why always the direction is from 5' to 3' direction? Because the component which synthesizes the DNA, who is our component? Our DNA polymerase. Our DNA polymerase which synthesizes the DNA always looks for a 3' OH. If it wants a 3' OH always, can it synthesize on its own? No. Whose help does it take? It takes a help of a primer. Who is our primer? DNA dependent RNA polymerase. And then what does RNA polymerase will do? It will lay a short RNA sequence, correct? No, it is a short RNA sequence, which you call it as an RNA primer. So what is it doing? It will give us a free 3' OH. Now, what will our DNA polymerase do? There is a specific DNA polymerase called as DNA pol 3. Our DNA polymerase 3 will synthesize our strand from 5' to 3' direction continuously. Now, I use the term called continuously. Now, let's look at it in detail. Okay, one second. Now, see, guys, our both strands were there, and the both strands they are attached with each other, and both the strands are parental strand. This is also parental strand. This is also parental strand. So, what happens to this parental strand? Helicase will come. Helicase is an enzyme. So it is an unzipper. Okay. So it is an unzipper. What does it do? Helicase will unwind the strand. What is unwinding? The bonds between these two strands are broken. So what bonds are there, guys? We know that these bonds are hydrogen bonds. So when you add water groups to this hydrogen bond, they get hydrolyzed and the unzipping will happen. So helicase came and it started unzipping. Where does it unzip? We know that it is not randomly unzipping. It unzips at one location, which is called as ORI site, or it is also called as origin of replication. Or you can also tell that this is a place where the replication even starts. Correct? No, this is called as ORI of replication. So in the ORI of replication or ORI site, what will happen? Helicase will see, okay, this is where I need to start the replication. It will hydrolyze the bond and unzip the DNA. Once the unzipping is done, our both the strands are unstable, guys, which we already know. Why unstable? The moment the helicase moves forward, the moment it goes forward, what will happen? Since these are, energies, right? So we know that the elements make bonds because they can exchange electrons and they're very unstable when they do not make bonds. So as the helicase moves forward, there is a huge risk that this unzipped DNA can rezip. Whatever we have unzipped now, it can come back together. So that is why what will happen to stabilize this, okay? Some special proteins come and these special proteins are called as SSBP. What is our SSBP? SSBP is called as single strand binding protein. Got it, no? So this single strand binding protein, what it will do, it will come and attach to this, the open nucleotides and it will stabilize them. The stability, how does it bring? It does not make them to go and rezip. Okay, it will not go make them reattached with each other. It will not allow them to reattach. So that is what SSBP will do. So once the SSBP has come in the place, now who has to come? As I told you before, we have primers which help our DNA polymerase. Who is our primer? Our primer is an RNA primer. Okay, our primer nature is what? It is an RNA. Who synthesizes this RNA? An RNA polymerase. What kind of RNA polymerase? It is a dependent, DNA dependent RNA polymerase. Clear, no? So till now, what has happened? We have unzipped and the bonds have been stabilized. Now, who is the second part of it? The very, very important enzyme that plays a role in synthesis of DNA is DNA polymerase. So there are three types of DNA polymerase, and if you see in prokaryotes, it is three types. If you see in eukaryotes, it is five types. But significantly, they are making the same function, and the role is very, very similar. So that is why we don't go deep into why the what is DNA polymerase in eukaryotes. Okay, we just understand this. Now you see, guys, in DNA polymerase there are three types: DNA pol 1 and pol 2 and pol 3. Okay. Now our DNA pol 3, okay, our DNA polymerase 3 is very, very important. Well, not all of them are important, but generally to remember, it's very, very important. Why? This is the one that synthesizes the DNA. Okay. This is the one that synthesizes the DNA. In what direction? 5 prime to 3 prime direction. It always synthesizes the DNA in 5 prime to 3 prime direction. So this is one function. What is the role of DNA polymerase 1? The role of DNA polymerase 1 is you have RNA, right? Already RNA, small pieces of RNA, even are you able to hear me, guys? I think my network got disconnected. Yeah, fine. Thank you. Now I think you can't see the screen. Let me share. Uh, Let me share the screen. Yeah, I think you can see the screen as well. Sorry for I think it got disconnected somehow. Chalo. Let's see. So we know that if I open the strand, it looks like something like this. You agree? This is the parent strand, and here is like it's wounded. Here it is unwounded, and here who is there? Here our helicase is there. Correct? No. Our helicase, what it is doing? It is unzipping in this direction. If the unzipping is in this direction, let's say that our one of the parent strand is 3 prime to 5 prime. Okay. And our other parent strand is what? From 5 prime to 3 prime. What did I tell? Always the DNA replication happens in what direction? I told that it is from 5 prime to 3 prime. So now what happened? Helicase came and it Sorry, people. Sorry. Somehow the internet is getting disconnected. Yeah, I know that it's very inconvenient. Okay, so maybe we have too much nazar in my classes. Sorry about it. So I hope you can hear me, guys, right? So now what happened to start the DNA replication? So an RNA primer, okay, RNA primase, okay, RNA primase will come, and this RNA primase will synthesize a short 10 base pair. Yeah, hold on. A short, sir, what does the polymerase one does? I'll talk about it. I'll talk about it. Okay. So now what will happen? A short 10 base pair of RNA, it will synthesize. So once it synthesizes a short, it's just like a push start. Okay. So once it does that, now what happens, our DNA polymerase gets all happy. So why does it get happy? Now it has got a free 3' OH. I told you. Once it gets a free 3' OH, now what this guy can do? Our DNA polymerase will synthesize a new daughter DNA in what direction? 5' to 3' direction in a continuous fashion. Correct? No. Now what it does, if you look at the DNA polymerase 3, okay, which DNA we are talking about? DNA polymerase 3. If you look at DNA polymerase 3, it has performed two functions here. What is the first function? You know that our DNA polymerase 3 will synthesize the DNA in 5' to 3' direction continuously. Correct? No? Continuously that it has done. What is the second function you can do? You can see that our DNA polymerase, it used the RNA primer. So this was the RNA primer, correct? Right? Do you agree? This is the RNA primer we started with. Now it expanded the RNA primer. See, extension of RNA primer, you call it as DNA RNA primers, we call. So here what happened? An RNA primer was already there. So to connect with the RNA, it has to go and attach to the RNA. So once it attaches to this RNA and starts synthesizing this new DNA, this process is called as DNA RNA primer extension. Okay, this process is called as DNA RNA primer extension, which is also the role of DNA polymerase 3. So what did we understand? In continuous strand, we have first the RNA primase, it will come, it will start synthesizing the 10 base pair RNA, and then this RNA is nothing but a primer RNA. Using this primer, a DNA polymerase guys, I don't know how much part of it is getting disturbed. Uh, Please let me know till where you heard the last, because it's getting disconnected very abruptly, which I'm not aware of. Okay, so let's go. Now we have to look at what is happening in the other strand. Now we know that the DNA synthesis happens from 5' to 3' in a continuous direction, but our helicase is moving in this direction. And the other strand's polarity, what is our other strand? Other strand is the lagging strand, or the other strand, the parent strand, which is having from 5' to 3' direction. Here, there is a new challenge. What is a new challenge? Every time a helicase moves forward, okay, a new base pairs gets exposed. And we know that the synthesis of DNA always has to happen from which direction? 5' to 3'. So the synthesis is happening in which direction? Opposite direction of the leading strand. So for that, what is the biggest challenge our DNA polymerase 3 has to face? It requires the 3' OH again and again, again and again. Here, once it is done, once it has put the 3' OH, continuously it can keep expanding. There is no doubt about it. But here, now listen here. If I put a primer here, let's say 5' to 3', you can clearly look at the direction, correct, right? You can clearly look at the direction. Now, what our DNA polymerase can do, it can use this 3' OH Correct? No, it can use this 3' OH and it can expand it. It can only do this process. So that is why in the lagging strand, the synthesis of DNA is not in a continuous fashion. So what happens? How is the process being adjusted here? For this, what will happen is our RNA primase has an additional rule. What is that? It does not lay the primer once. Instead, what it will do is it will synthesize 1, 2, 3, 4. Like this, you have this RNA, 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 RNA. Got it? So these are called as RNA primers. Okay. Who synthesized this? RNA primase synthesized this. Now in what direction? 5' to 3', 5' to 3'. Okay. You can see that it's like a tiles, no? Like steps. 5' to 3'. Like this, it has put small primase in the lagging strand. Now what it will do, once this primase is available, you can clearly see that there is free OH group here and there. There are free OH group in the 3' end. Now, what is the advantage? Who gets the advantage? RNA polymerase 3, guys. So, what is our RNA polymerase 3 will do? RNA polymerase 3 will expand this 5' to 3' gaps. You can see. So, what do you call this? RNA, DNA, RNA, DNA, RNA, DNA. So, does it mean this DNA, RNA primer extension? Is it doing in the lagging strand? Yes. Now, can we have RNA in our DNA? No, we can't have RNA in our DNA. Very strict no. For that, what happens here? Our DNA polymerase 1, our DNA polymerase 1 will come. The role of DNA polymerase 1, the first function is it has an endonuclease activity. Can you guys hear me? You guys are in the class. I don't know, like if you're following me, like if you missed out something, please text in the chat box. Okay. So I was saying that in nucleus, there are two types, exonuclease and endonuclease. And if I talk about exo means what? Corner. Okay. In genetics language to understand. Okay. What is endo? Endo means inside or in the middle, I can tell. So if our any enzyme or any protein, if it has the capacity to cut in the corner, it is called as exonuclease. If it has the capacity to cut in between, that is called as endonuclease. Now, what is our RNA polymerase 1 do? RNA polymerase 1 has the capacity of exonuclease activity. What it will do is it will remove these RNA primers through exonuclease. Okay, it will start removing this RNA primer. So let's start removing this RNA primer. Okay, who does this? Oh, sorry, I removed everything. Okay, DNA polymerase 1, it has removed all the RNAs. Okay, even this primer is removed. Now, what is the role of your DNA polymerase 1? First one is it removed all the RNAs through exonuclease activity. Okay, now what it will do is it will fill this gap with DNA. It will synthesize a short DNA. Okay, it will only synthesize its short DNA. Now, replacement of DNA with RNA with DNA is done through exonuclease activity. What is the second activity it will do? Now the gaps are there, right, where the RNA was. That gaps has to be filled by who? The DNA pol 1 only. So this gap filling is done by DNA pol 1. Okay, so that is called as what? Creating DNA primer. Okay, this is called as creating DNA primer. Got it? So replacement of RNA primer with DNA primer is done by who? DNA polymerase 1. So once this is done, you can clearly see that here there is a gap. Can we have gap in the DNA, guys? No. Here there is a big gap. Here there are small gaps. So whenever you see a long thread with small pieces, what do you call this? You call it as fragments, correct? No, you call it as fragments or fragmented DNA. Can we have fragments in DNA? Definitely no. So when we see a long thread with small pieces, what do you call this? Fragments. Dear students, I know that it is very frustrating to have a you know a connection problem. Please let me know your thoughts, like how far are you able to cope up with this? And hopefully, this is the last class that I will be taking from the laptop. Hopefully, so I will get back to the classes. Because of the bad health, I'm taking from home. I mean, like in the laptop. So really sorry about it, about the network issues. Clear? So I hope the last thing that you heard is DNA ligase. Hopefully that's what you heard. And if it's not, if it's somewhere you feel that it is discontinuous, please let me know. Okay, please let me know. So your communication is quite a little bit important right now because I'm in the shadow, which I don't know what's happening. So no. So DNA ligase, what it will do, it will start sticking up all the Okazaki fragments and it will make it a continuous DNA. So we have understood the role of DNA Pol 1, Pol 2, and DNA ligase, helicase, SSBPs, and the RNA primase, etc. Okay, so the whole process requires what? It requires energy, and the whole process should be damage free. So that is the whole idea behind it. Correct? No. So it should be a damage free DNA, and you require energy for the whole process. So first we'll talk about the energy. So in biology or in chemistry, you know that the reactions simply don't happen. It requires investment of energy. And we know in all the biological organisms, the energy is stored in the form of chemical bonds, correct? No? We have bonds. So what kind of bonds? Our most energy currency is ATP, which you are very familiar with. So ATP has triphosphates, and the energy stored with the covalent bonds that adenosine makes with the three phosphates. Okay, so whenever you break the phosphate group, a huge amount of energy is released. So similarly, we have other energy currencies for the DNA replication, which is called as ATP, GTP, CTP, and TTP. Okay, so these are all currencies of energy. So now what will happen when they come for the assembly? Their bonds get hydrolyzed and the huge energy is released. So who does this job? The job of using this energy, breaking their bonds and using their energy is nothing but DNA Paul. Okay, our DNA polymerase has a capacity to use them and break their bonds and use that energy to construct a new DNA. So what are these? They are nothing but deoxy ribonucleotide triphosphates. So these DNTPs are the ones they come and they become monophosphate. For example, adenosine monophosphate, gynosine monophosphate, cytosine monophosphate. They turn into triphosphate to monophosphate. When they become triphosphate to monophosphate, a large amount of energy is released, and this energy is utilized for the synthesis of DNA. Okay, so that is the energy. Next one is no error. Okay, you need not have, you shouldn't have even a single error. So you can understand how big is this process. Each cell, 6.6 billion base pairs of DNA. I told you, you have around 30 trillion cells. Look at the amount of error freeness that we have in our whole synthesis of DNA. Correct? No. So no error is brought by who? It is first brought by DNA called 2. Very, very important function, DNA polymerase 2, which we don't give the respect at all. See, proofreading means every single base pair has to be seen, adjusted, made corrections. If it is wrongly based, if it is wrongly paired, yeah, please tell me. Yeah. If it is wrongly paired, you have to readjust it. Okay. And if there are nicks or any breaks in the DNA, it has to restitch it. If there is any bubbles, it has to readjust it. So all this is done by DNA polymerase 2. The repair mechanism is more complex than what you see in the whole replication. That's why they have not touched the DNA polymerase 2's repair function at all in your textbooks. Okay. Hopefully you'll read in the future. And what is the second thing? When the helicase is unwinding the DNA, okay, you have a special enzyme called as protein, okay, topoisomerase. So topoisomerase or gyrase in eukaryotes, topoisomerase in prokaryotes, okay, in general. So the role is very, very similar. So what you can see is before the helicase, if the helicase is moving in this direction, if this is the double bond or this is the double helical structure of your DNA, when it moves like this, there is a stress called as torsional stress. Okay, it is called as torsional stress. So this torsional stress, what it will do is it will lead to supercoiling. Okay, supercoiling. What is supercoiling, guys? So in very, very general terms, if I take my two threads and if the two threads are interwound like this, and if I start ripping them apart or separating them apart, you can see at the back end, it is getting lumped or it becomes like a ball or it becomes supercoiled. So this is very, very natural. So this torsional stress leads to supercoiling and supercoiling leads to what? Breaks. Okay, it leads to breaking. So to avoid this, what it will do is the topoisomerase, if this is your double-stranded DNA, this is the helicase. No, this is the helicase. It's moving. Let's assume the topoisomerase. What it will do is it will break the DNA here and there. It will make small, small cuts. So when it makes the small, small cuts, so it will see the helicase is coming. It has made a cut. Now what it will do as it's coming closer, it will stitch back. Again, it will go further. Again, it will make a small cut. Again, it will close the cut. So it go further, make a small adjustment in the. It will break the whole DNA. It will nick. We call it as nicking. Nicking means what? Small cut. Okay. A nicking is done by who? Topoisomerase. Not where. If this is the helicase, far end from the helicase, what it will do? It will see the helicase coming. It will decide, okay, now there is a stress buildup. Okay, cut the DNA. So when the DNA is cut, the stress is reduced. Okay. As the helicase is coming closer, stitch back. Then again go further. Again, you can feel the stress. Again, cut the DNA. Again, you see it coming closer. Stitch it back. Again, go further. You can see here, right? So this is called as avoiding the nicking of DNA. So nicking of DNA is avoided by topoisomerase or gyrase. What does it do? It will relieve a stress called as torsional stress. So what does torsional stress leads to? It leads to supercoiling. Okay. So how does it relieve supercoiling? By making nicks in the DNA. So it makes small, small nicks in the DNA further to the helicase so that the DNA will not get broken. Okay, or supercoiling will not happen during the replication process. Likewise, you can see that both your leading strand and lagging strand by the end, you can see the terminal end. That means that the corner, the end portion of the DNA, until the end portion of the DNA, this process will continue. Finally, it results in separation of two parent strand. I mean, like two daughter DNAs. So this will become one DNA. This will become another DNA. So this is the end process. You understand my point? After they reach the end point, all of these enzymes and proteins, they disperse or some will get denatured and the other will go for another process. Or if they're assigned by the cells, they will go for another replication cycle. Okay, so this is about DNA replication. In prokaryotes and eukaryotes, the process is absolutely same, but the only difference is the speed of replication. The speed of replication in prokaryotes is quite faster than in eukaryotes. The reason being, the eukaryote DNA is quite well packed, okay, whereas the prokaryotic DNA is very loosely packed and it is very, very abrupt. So another important difference between prokaryotic replication and eukaryotic replication is you can find a lot of errors in prokaryotic replication, but in eukaryotic replication, it is highly proofread. That is why we are error free. Okay, most of us are error free, blissfully. Anyways, guys, do you have any doubt? Okay, now it is open for you. Tell me if you have any doubt, guys. Yes, no, you have to respond, guys, a little bit. Now, this is a very interesting concept which I like to discuss. Tell me. Okay, thank you for the response. Only Sneha response. Other guys, what happened to you? I think we have Bharat, we have people from Shillong. I don't know your names, guys. I will get to know soon. Okay. And then Guru Bharadwaj is there, Basavraj, and we have Keshav. What happened to you guys? Are you here? Say hello at least. No. Now we have to go for mechanism of transcription. So the whole idea, not the idea, the whole reason why the DNA exists itself is to give orders. It's like a give, it is a supreme. So it will tell what to do, what not to do, how to do, how to becoming you. Okay, becoming an organism, becoming an expression. Crazy, something that you it's very deep if you understand. Okay, so how does it do? How does it manage this whole process? The management department is very simple. I give the orders and you have to assemble something that I need. As simple as that. The same chain of commands is also followed even at molecular levels. So we know that our DNA carries important information in the form of what genes. And this genes is nothing but something which is functional. And what happens to the rest of the DNA which is non-functional? The non-functional DNA is very, very high. In every organism, the amount of non-functional DNA is too high, guys. So only a little portion, let's say that less than 2%, okay, less than 2% of the DNA is your genes. 98% of the DNA that you have is not at all functional. Some people call it as junk DNA. Some people call it as useless DNA. Some people call it as structural DNA. Some people call it as ancestral DNA, which we are not worried about. So what happens to this one that is functional? The functional DNA or the genes has to make sure that the cell constructs what it requires. So for that, it needs to maintain a chain of commands. The first way the DNA maintain its chain of command is through a process called as transcription. So what happens in transcription? We have the DNA. The DNA is transcribed into a message, okay, in the form of an RNA sequence. An RNA sequence. So this RNA sequence construction using a DNA, so this process is called as a transcription. Now, we know that each DNA is composed of two strands. So we have two strands in the DNA. You can see here, as usual, there are how many strands? There are two strands. And each strand has its own polarity, right? So we know that one strand goes from 5 prime to 3 prime direction, you can see here. Other strand goes from 3 prime to 5 prime direction. Now, does the RNA transcription happens in both the strands simultaneously? The answer is no. So if you have two strands, only one of the strands is participating in the process of transcription. So what strand are we talking? This is a DNA strand. This is a DNA strand. Both of them are the part of original strand. So they both are connected with each other. Correct? No? So one is 5 prime to 3 prime. Other one is what? 3 prime to 5 prime. Now my question is both the strands involved in the whole process? My answer is no. Only one strand is involved in this process. What is the name of this strand? So this strand is called as the coding strand. Wait, we have two strands. I will call it as template strand You will understand. Template, and then you have something called as coding strand. So don't get confused with the names. What is a template? Template is something, for example, if I ask you to write an essay, if I ask you to write an email, if I ask you to write a formal letter to the editor, or if I ask you to make your own resume or biodata, what you will do, guys? You will ask me, sir, do you have a sample? Do you have a template? So that's what is happening. So the template is your DNA that helps in the manufacture of RNA. So your template, let's say that this is from 3 prime to 5 prime. So this will be our template strand. How did I say that this will be my template? It's because it's going from 3 prime to 5 prime direction. Why? Even the RNA synthesis will happen in what direction, guys? 5 prime to 3 prime direction. Even the RNA synthesis is always happening from what direction? 5 prime to 3 prime direction. So that is why if my RNA synthesis is from 5 prime to 3 prime direction, then my template DNA will be in what direction? It will be in 3 prime to 5 prime direction. Correct? Now what happens to the other DNA? The other DNA is going from 5 prime to 3 prime. No. What do we call this DNA? This DNA is called as coding DNA. Now, I don't know. Sir, coding means something that you code. Template means copy. Why did we give this word called as coding strand? Which is none of our concern right now. So what do we have here? We have our template strand, and our template strands polarities from 3 prime to 5 prime direction, and our coding strands polarities from 5 prime to 3 prime direction. Guys, I know that this is a very simple information for you to remember, but just observe. Many students make a big mistake whenever they ask you to write the structure of the gene. Okay, whenever they should give this picture of the RNA transcription, okay, or the transcription, what you guys do, you will forget what is the polarity of the template strand, what is the polarity of the coding strand. The template strand's polarity is always from 3 prime to 5 prime direction, and the coding strands, polarity is always from 5 prime to 3 prime direction. Now, what did we understand? We understand that the functional part of the DNA is called as a gene, and the gene is where you have all the information hidden for the expression of characteristics. So for the expression of characteristics, there is a chain of command, or that there is a chain in which the information passes. And that is nothing but the DNA has to be transcribed, or the first message comes through the manufacture of RNA. So how does the manufacture of RNA or the synthesis of RNA will take place? It will take place in the DNA strand. So here in the DNA strand, we have two strands. One strand is going from 3 prime to 5 prime direction. Other strand is going from 5 prime to 3 prime direction. So the synthesis of RNA is always in the forward direction. What is the direction? 5 prime to 3 prime direction. If my RNA has to be synthesized in 5 prime to 3 prime direction, so the strand it uses for the synthesis of RNA is what? It is a DMA. Correct? No. So what is the polarity of this DNA? It is from 3 prime to 5 prime. What is the name of this DNA? This DNA is called as a template DNA. Whereas what is the opposite strand called as? It is called as coding DNA. Got it? No. So again, the whole synthesis of the RNA is an enzyme enriched process, I can tell. A lot of proteins, a lot of assembly of units has to come to make sure that this particular process is quite successful. Hold on. Sir, what is the role of the template strand? Of course, see guys, this is your DNA. Okay, let me explain. This is the question I got. Okay, one of your friends has asked me a question. What is the role of the template strand? Okay, sure. Now I have two DNA strands. Now this is my template DNA 3 prime to 5 prime. Okay, now this is my coding DNA 5 prime to 3 prime. Now my RNA synthesis has to happen here. Okay, let's say that A, C, C, G, T, A, G, let's say. These are the codes in the DNA of my template strand. And even in coding strand, its complement code will be there for A, T, for here T, here G, here G, here C, here A, here T, okay, here C. Now what will happen? Here, I told you that here the synthesis will of RNA will happen. How does the synthesis will happen? By complementation. Now, in simple, what is transcription? Manufacture of RNA. or making of RNA is called as what? A transcription. From DNA to RNA is called as transcription. How does this process will take place? To convert the DNA into RNA, we don't convert DNA. We use codes. Okay. Codes that are in the DNA are read into an RNA form. How? For example, you know that in RNA, we have A, U, C, G. A, U, C, G are the base pairs. Or let's complement here. For A, if I have to construct RNA, what will be here? U. Here, U. Here, G. Here, G. Here, C. Here, sorry. Here what? Here it is A. Sorry guys. Here it is what? Here it is again U. Here it is what? C. Now this is what? This is your RNA. Got it? What is the function of the template strand? Template strands anti-code, okay, or complement. See, for A, what did you do? You complemented with U. What is this U, 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 G, G? This is nothing but RNA. To get an RNA, you need to use the template strand. So the codes in the template strand are read into the codes of RNA. Okay. So template DNA's codes are read in the form of, or read means reading, or synthesized, you can tell. The codes of the template strands are synthesized in the form of RNA. How does the synthesis will take place? So here is my base, which is, here is the code that is present in my template strand. What do? I will construct a complement code for A, U, G, C, G. So by this, so this is my information. This is my crucial RNA that helps in the rest of the things that I have to do. You get my point? Did you understand? So in simple terms, what I can say is the role of the template strand is nothing but it gives a reading frame. Okay, it gives the reading frame for the construction of new RNA. Did you understand? I think who asked the question? Mamami Devi? Momoni Devi? Sorry, Momoni Devi. If I pronounce your name, please forgive me. Wrong. Okay. If I pronounce it wrong, please forgive me. Did you understand? Please, can you confirm? Yeah. You can put a message. Okay. It's easy for me rather than seeing the. Okay. Chalo. Uh-oh. Got it, no, guys. So we understood the basic. So this is where the DNA transcription starts, and sorry, RNA transcription starts. And I told you what is coding strand, what is template strand, and everything. Now, let us talk about the process of transcription. How much time do we have? Yeah, 8 minutes. Let's see. So the process of transcription, again, your DNA, both the DNA are tightly wound, correct? No? So again, this has to be what? It has to be unzipped. Unzipping has to take place. For unzipping, who is helping? Our DNA helicase? Here it is. Our DNA helicase will help in the unzipping. Okay, it will unzip. Second, what it will do? It will have to make sure that the synthesis of RNA takes place. So for the synthesis of RNA, who is helping us? RNA polymerase is helping us. Like the DNA polymerase, we have RNA polymerase. Okay. So these RNA polymerase, okay, are very, very important for this. I mean, like they do the whole role. Okay, they do the whole function of synthesis of RNA. So in DNA polymerase, we have several types. Okay, DNA pol 1, DNA pol 2, DNA pol 3. Okay, sorry, sorry guys, RNA pol 1, RNA pol 2, RNA pol 3. Okay, so that is RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Like that, we have three different polymerase. Now, let us talk about your RNA. Okay, RNA polymerase. RNA polymerase is very, very important because each of the RNA polymerase, they have a specific role. Okay, so the role is, excuse me, the role of this RNA polymerase is to synthesize different. Oh, sir, why coding strands take part in the transcription only? No, template strand takes a role in the process of transcription. Okay, but the coding strand will not take place. See, guys, simple. You have two strands. One is 3' to 5'. Other one is from 5' to 3'. I told you this is template. I told you this is coding. This template strand is participating in the process of transcription. The coding strand is not participating in the process of transcription. Why is the question? The question is very simple. If I have a gene, okay, let us talk about your sickle cell anemia, guys. In sickle cell anemia or thalassemia, any genetic disorders that you learn, you understand that every single code that is present in the DNA makes sense. It is very, very important. No, in the gene, it makes a lot of sense. So imagine if this is my template. Template may I will say that this is the code that is present in my template. Okay. What will be the codes that are present in my uh, other strand? For A, what is in the other strand? T. Here T. Here G. Here G. Again here C. Again here C. Again here G. Now, what did you understand here? Here, this is one code. Now let's try to decode it. Okay. Why it is not the same? Two reasons are there. First reason is that always the synthesis of RNA takes place from what direction to what direction? 5 prime to 3 prime direction. So that is why your coding strand will not come into play. So that is the first reason. Now, if I have two strands and if my strands is AACCGG, okay, let me call this TTGGCC. So this is template, this is coding, guys. Now see, if I synthesize RNA from this, what will be my RNA? UUGGCC. Okay. If I synthesize RNA from coding, what it will be? It will be AACCGG. Now, are these same? No, definitely no. You know that even a small change in the single base pair can completely abrupt, can completely change what it means or how it means or what it becomes. So that is why only one strand can act as a sense strand and the other strand is a nonsense strand. The sense strand is your template DNA. The nonsense strand is your coding DNA. Okay. If a gene is coded in the DNA, it is coded onto the strand which gives rise to the RNA. That strand is called as a template strand. And the other strand, the coding strand, you can see that the codes become an anti-code and they're not same. The expression is not the same. Did you understand, sir, why coding strand take part? No. You understood now? Not coding strand, it is template strand. And I gave you a reason. I understood your question. Why coding strand takes place in transcription only? Your question should be, sir, why the template strand only takes place in transcription? Now I told you the reason. First reason, always from 5 prime to 3 prime. Now you understand. If the codes are there in your DNA, you cannot have an anti-code and expect the same result. Okay. You, as we move further, we'll understand how specific they are wired, how important that each base pair is. Okay. As we move on, we will understand. Anyways, it's only 3 minutes left, and I will not. I don't like to rush. When I rush, things will go wrong. I hope that you understood DNA replication. If you have any doubts, you're always free, guys, to ask me. And one second. So as usual, you have a role to play. See, your feedback gives me a lot of insight to how the class is going and have you understood or not, okay? Or if you're facing any trouble, all that I will understand. So in the last class, I understood many of you were dissatisfied with the DNA transcription. So that's what encouraged me to uh, revise it again in spite of losing a class. I don't mind. But the first priority is I want you guys to understand, okay? Yeah. All right, guys. Some people still feel the difficulty. I understand through your polls, and I don't mind. I definitely respect your honesty and your opinions. And this is not sufficient. Why? Because I hope somewhere there I am trying to do the best, but sometimes what will happen is I will not understand where it is lacking. Is it the language, or is it the words, or is it the delivery of the concept? So what you have to do is either you ask me to repeat, or you tell me in a certain sense that okay, this is the way I will understand. Okay, so that will help me, guys, even to make it better. So anyways, thank you for your feedback and your honesty, and have a nice day. I will see you guys soon. And hopefully we'll have the better sessions. Okay, have a nice day. Thank you. And please share your opinion whenever you want. Okay. Excuse me.